This is Robert Kraft, and I'm your host for SNN Network. And joining me today is Dr. Anil Diwan. He is the president and chairman of NanoViricides, Inc. It's a publicly traded company. The symbol is NNVC on the NYSE American. Dr. Diwan, great to see you. Thank you for joining me today.
Nice to see you again.
It's been a while. It's been a while, that's for sure.
Yeah.
Well, like I said, it has been a while since we've seen you and had you on to do an interview. So before we get into some of the company updates, which, are, I'm very much looking forward to digging into, let's start with a quick overview and history of the company, and then we'll go from there.
Yeah. So basically, this company was founded in 2005 with Dr. Seymour, who is an HIV expert, as you know, and he wanted to work in virology. I had developed the nanomedicine technology since 1992, and he saw the opportunity for taking that further into drug development space. So we started talking to each other since 2003, 2004 timeframe, and then eventually we formed a company, took it public through a reverse merger so that we could raise money. He raised initial monies, quite a bit of small amounts of money initially, and then as we started showing data, how the stock market behaves, so we were able to raise more monies. And so initially from about 2005 through 2015, what we were trying to do was we were trying to show that this novel concept that we are working on does actually work and does have a place in drug development. And so we went after many different projects. You are aware we started with influenza and H5N1, and we actually went to Vietnam to be able to do work on H5N1, and we saw success there. We saw success in animal studies. Then we switched to HIV. We also did dengue, Ebola, other projects. All of these projects were basically to get attention. At that point, we were working out of a small lab. We did not have characterization. We did not have manufacturing capabilities. We had very short staff. And so because of that, it was mainly so that we could get some grants or we could get additional, we could get further with the projects. And so that went on. We almost got a grant from DOD for dengue, and it went to the contract office, but it did not materialize. So eventually we had to restructure our own thought processes. And what we decided to do is we will work on what we can do ourselves with our monies. We knew by that time how much money we could raise on the market, on the stock market through equity raises, and what kind of expenses we had ahead of us. So we based the projects on that. So we literally put HIV and influenza behind on the back burner, and we started working mostly on herpes simplex viruses. We had great data on HSV1 in animal studies. We have that drug in development. And then even from that, we switched further to a skin cream for shingles rash, because we saw a great opportunity there that there is no drug there. There is almost everybody who crosses 50 is going to get shingles at some point. Now, of course, there is a GSK vaccine that was introduced a year ago, and that will cut into something, but it is only being produced 7.5 million doses a year. So there is plenty of opportunity for the market in there. And so we pushed on that. Around 2015, we built our modern site, which is a pilot manufacturing capability, multi-kilogram quantities. We have up to 75-liter reactors here now. And we have CGMP quality or CGMP capable facility. We have quality instrumentation setup. We have quality analysis setup. And we have nanomedicines characterization, one of the best characterization facilities, if I may say so. The only thing that we do not have for nanomedicines characterization is SEM and TEM, electron micrography, which we don't need because our materials are soft materials. They are soluble in solution. And so they are not hard particles. So that's what went on through 2015. In 2015, then we focused, after we had the facility, started making larger materials, batches, started getting characterization data together. And 2017, we started our tox package studies for our first drug candidates for shingles. 2018, that study was almost complete. 2019 actually is when we did the the GLP safety toxicology, the final portions. And now we have been writing the IND application. So that is good. Then in the process, as we were doing the IND application, this thing hit us in, the December new cases that had come up. And fortunately, we had worked on MERS coronavirus before in 2014. Those candidates did not get to testing because it was a public lab outside of United States that was going to do the testing, and they lost interest because of the Ebola epidemic, that outbreak that came in at that time. So, but we had done the work. So we had some starting position, and we did some initial work on modeling, and I'll get to the technology in a minute. I'm just trying to give you corporate history right now. Yeah, so we did some initial work on modeling, and in January itself, we already had a candidate that we thought was very good. And then we put in 2 or 3 additional candidates. We very quickly, we set up testing capabilities in our lab for coronaviruses that we can use in BSL-2 conditions, biological safety level 2, because we don't have a BSL-3 or 4 lab. Those are very regulatory-wise, they're very expensive to run, and a lot of bureaucracy also is involved in terms of record keeping and things which we cannot handle at this time. So we had kept our lab to be BSL-2. And so these are coronaviruses that infect humans seasonally. About 50% of the colds, common colds that we get, and respiratory, upper respiratory diseases typically are from coronaviruses. So we are getting them all the time. So 2 of those viruses we brought into the lab. One of them uses the same receptor as the coronavirus that is causing the major outbreak and pandemic now, the SARS-CoV-2. So that coronavirus is called NL63, NL63. And we have been using that as the model virus for doing testing for our candidates. And we have been developing candidates. You probably saw our news release today morning that we have actually alluded to this in the Planet MicroCap presentation, that we already had data, and I'm just waiting for verifying the data. One more study is going on in our lab, and once that is done, we'll be able to talk about it. So that we have those data sets now, and we have confirmed that we have very strong activity against these 2 different coronaviruses, which are very, very different. And you can read the press release that has more details on it. And what that means is that these candidates will continue to work against the SARS-CoV-2 as well if they work in the first place, but they will continue to work even as it changes genomically, as it mutates, and so on and so forth. That's very, very important, because with antibodies, you cannot have that guarantee. With vaccines, you do not have that guarantee. So we need something that will continue to work as the virus drifts. As you already know, the virus has been drifting, and that is going to happen. It's a natural process for RNA viruses. Fortunately, the coronaviruses do not drift as much as influenza and HIV do, but they do drift. And you are seeing that in different kinds of pathologies that are being seen in the world. In China, they had described 2 different pathologies, 2 different rates of fatalities in the, and they claimed that the first one was less pathological, meaning fatality rate was low. The second one was higher in fatality rates. In the United States, across the 2 coasts, we are seeing a distribution of different kinds of diseases actually being caused by the same virus. And we are seeing different kinds of diseases in different people in the same areas also. So it's likely that there are many different kinds of small changes in the strain that are circulating. And obviously, we don't know definitively until all of the sequencing data and those research reports come out. But it is very, very likely that that is the case. And so because of that, developing broad spectrum agents against viruses is very, very important. So now I will get to the technology. How do you think we did it? You have to find something that the virus is not changing, no matter how much the virus is changing. Right? So that's what you have to do. So in the case of coronaviruses, for example, in this case, SARS-CoV-2, it is going to bind to the same receptor on the cell surface, ACE2, ACE2, angiotensin converting enzyme 2. That's a specific receptor that the SARS-CoV, original SARS coronavirus bound to. That's the same one that this coronavirus is also binding to. And the NL63 that I talked to you also binds to the same receptor. So that's why we use that as the model. So when we do molecular modeling, we had molecular models available of the SARS-CoV, the first one, and it's binding to ACE2. And what we do is we try to mimic the ACE2 portion, the cell side portion, where it is binding to the virus. So that is the invariant site. The virus changes as much as it wants, it still binds to the same site. That has been very well known. It's very well established now. And so that is what we try to mimic. And then typically what happens, and you probably have seen these things in the SARS-CoV drug development also, there is a report now which actually substantiates our work, that they have developed soluble portions of ACE2 receptor itself, put them in mice, and have been able to clear the virus. So typically that's what people try to do. That doesn't necessarily work in the clinic. It did not work in the case of HIV. It has not worked in the case of many other viruses. So we go one step further than that. And the reason for that, why it doesn't work, we think, is because the virus itself is a spherical or oblong particle. It has many, many different points by which it can bind to the cell. You see the coronavirus cartoon, you see a lot of different those club-shaped things. Those club-shaped things are the ones that are binding to receptors. So I'll get back to it. So what happens is that the virus binds to the same cell surface receptor in the same point. And that's why we try to mimic that. And there is substantiation of our approach now. What has happened, people have tried the soluble portion of ACE2 and shown in mouse model that it does work in neutralizing the SARS-CoV-2. Now that doesn't necessarily work clinically, and it has not worked clinically in many other cases. The reason that it does not work clinically, and this is what I was trying to explain that time, is if you look at the SARS-CoV virus, or any virus, it is a relatively rigid sphere, and the club-shaped spikes that you see, it's called a spike protein, and that is what is binding to the ACE2. So as you can see, all around the ball of the virus, there are many of these spikes that are binding, have the ability to bind to the virus. So if you put in a few of those soluble proteins or portions, then they are only coating a few of those things. There are still many open that the virus can interact with the cells with. So that is what happens clinically. You can't raise the amount of proteins in your bloodstream and more in the lymphatic systems and things to the levels that they need to be effective. So what is the solution for that? What I had come up with long time ago in 2005, that is when we formed the company, and 2003 is when I had done the first patent that we got approved in the United States. So what I had come up with is that what you need to do in this kind of situation, you have a rigid ball. You don't want another rigid ball interacting with it. It's going to be a tangent interaction. What you need is something else that's going to be able to encapsulate it. So sort of like the Venus flytrap flower that we talk about, that just it attracts the insect and then grabs it and closes onto it. So that's the kind of material you need. And we believe that we have actually made that happen. When we started, it was called science fiction, and even now people describe it as, oh, that is like science fiction, but we have data now and it works. So we have very strong data, as you saw, even on the SARS-CoV-2. We have sufficiently strong data, also safety evidence that we are taking it to IND for shingles, and then we'll be going into the HSV1 and HSV2 with the same things. So herpes class viruses. So this is a way of generating things against viruses that the viruses cannot mutate against, and they are not dependent upon the immune system functioning well. In the case of SARS-CoV-2, it is very well known now that yes, you get the infection, you get very high levels of viral load, but what is killing you at the end, with the people that are dying, they are getting killed by their own immune systems. So the immune system dysregulation is one of the things that viruses do. And that is why they don't get regulated. The virus doesn't come under control. And it is important that you don't depend upon the human immune system to attack the virus. And that's what we believe we have achieved too. Antibodies require the human immune system to be functional. There are two parts to it, something called complement system in the bloodstream, and then the cell system, the cellular system, dendrites and other cells that in the lymphatic cells system, that the antibodies will basically label the virus at few points, and then they will depend upon these systems to clear the virus. What we believe we are trying to do is taking that whole involvement of immune system away and build a nanomachine that is going to attack the virus, encircle the virus, uproot the GPU, the glycoproteins with which the virus binds to the cellular receptor, so that the virus becomes non-infectious, and then it becomes fodder to whatever in the body for clearance.
Got it. All right. So that was a lot. I think we can. I think we can all agree there that you know, I feel like I just actually sat through one of my my AP bio classes back in high school a little bit. But you know, in all seriousness, you know, you already kind of spoke to one of my next questions, which was where some of these drug development programs are. Mm-hmm. More or less, I think we can agree that all your programs are still preclinical and not in one way or another in the pre-IND, IND phase, correct?
Right. Okay. So shingles is the most advanced. It's we are writing the IND application for it. So all the data is there. Yeah.
Okay, got it. All right. So I'm gonna go to my next question, because I think this was one of the main reasons I really wanted to speak with you guys today, because you know, we've known NanoViricides for a while now. I've interviewed the firm
since almost 2005.
Yeah, for a while. You know, I mean, I've interviewed the firm's various CEOs over the years. You know, and the company, you alluded to this during your opening answer there, that the company has announced over the years its intentions to develop drugs to combat Ebola, HIV/AIDS, and now COVID-19. But seemingly, each time it gets held up in the IND preclinical stage. Now I asked my following question, keeping in mind that getting a drug to clinical trials, let alone approved, is not an easy task and takes a lot of time and money. However, you know, with you at the helm, how is the development for a drug to combat COVID-19 being handled differently than say some of the other indications the company has gone after over the years?
Yeah, so we have been very lucky in this situation. I already alluded to that we had done work on the MERS-CoV and SARS-CoV before. So we had things in the library, in the virtual library, and some of them were actually synthesized materials already in our hands. You know, so because of that, we were able to get a very quick jump start. Most of the workload in drug development is basically drug discovery takes a very long time initially. And then the rest of the time is you are just working on getting the drug through into the regulatory practices, getting the manufacturing set up, and so on and so forth. So what has happened in this case is that the drug discovery took literally maybe a week, you know, because we had them in hand. And then the synthesis was already established. Manufacturing, we had already gone to kilogram scales. The CMC was written up. The polymer side CMC, we are literally copying. And the ligand side CMC is half written up already. So most of the work that we did along the way is already benefiting this program. And that's why we are very, very close to being able to get to the next step. Now, what is needed for getting to the next step is, I again talked about that also, that the drug candidates have to work against SARS-CoV-2. So that collaboration is in progress right now. We are hoping that we can announce that very quickly, very soon. The second thing that we have to do is hopefully we can do some animal model studies. There are no true animal models for SARS-CoV-2 yet. The developments are going on, but if we can get there, that will be great. If we can't get there, that is not really a showstopper at this point. And then the third thing we have to have is establish the safety of these drug candidates, you know, in small animal models, and then sufficient data to be able to give confidence that we can go into first in human studies. And then we believe that we will be able to do the standard toxicology package along the side because of the urgency and the emergency situation that the whole world is facing. So there are things that need to be done. There are things that need to happen from the regulatory side, and there are different thought processes going on. If you see what happened in COVID-19 space to date, is that only the drugs that had gone into clinical trials before for other viruses or approved drugs for other indications have gone in clinical trials. And that is because any new drug that you develop, you do have to go through safety establishment. First, do no harm is the principle. So we have to get to that stage. That study is going to be commissioned pretty soon. Initial safety studies. Fortunately for us, because of the way we have built the polymer based on polyethylene glycol, PEG, which is used for making antibodies safe. That's the basis of our polymer. So we usually do not see any safety toxicology issues. And so far we have not seen. But the ligands are different. And if there is a toxic ligand introduced, then of course you will get toxic results. So you do have to go through the studies. So that's going to be the one thing. Fortunately for us, when we did the shingles full tox package, the data that we collected in the preliminary small animal study and what we collected in the full tox package were essentially similar, very similar. There were no surprises. So that gives us more confidence that the small animal study alone in safety toxicology is going to be predictive of what will happen in larger scale, the full-fledged toxicology. But there is work to be done.
Yeah. Right. Oh, no, absolutely. I mean, look, here's really, I think, the biggest fear I think a lot of investors have, because when it comes to nanoviricides, it's the same things that I've observed over the years. So just wanted to quickly follow up on this. I mean, it has to do, I feel like, with focus, and the fear that, God forbid, there's another pandemic or there's another, Ebola-like, disease that's now going to come out here that, look, it's going to take a long time for to get where you guys are today, to get to that treatment for COVID-19. So, what's being done from a corporate level, from with you at the helm, to make sure that if something else does come up down the road in a few months from now or a year from now, that still the focus for the company is going to be on the indications that you already have been publicly talking about?
Right. So that's what I was going to say to you, that we have a corporate business plan, and that is based right now on herpes viruses, HSV1, HSV2, and shingles. And there are more than 8. You can look at our 10K filings. There is a detailed description in there. More than 8 indications that we can go on those multi-billion-dollar space that can keep us busy forever, right? So because drug development, each drug just to get into IND stage, it's a 1-year work, even if we are modifying from current drugs candidates to other ones. Okay, so that is going to be, that is not, we have not taken our eyes off of that. The IND write-up is going on. We are doing this mostly because we were very lucky, like I said, that we had things in hand, and all the data and information that we are developing currently in this scenario is still also helpful for our other programs. So that way we are not losing anything either. We are losing, I would say we are losing time and money, yes, because we are defocusing to some extent. But it is, as you know, it is a humanitarian need right now. Everybody is working on it, and somebody needs to be successful, right? So we believe that we have a good chance at success. We have seen that in cell culture studies now, and we know it is these candidates are safe from other studies. So of course, we have to prove that for this particular situation also. So that's going on. We are not taking our focus away from the shingles program. The clinical trial design is going on. As you know, COVID-19, the whole the movement of the pandemic through the population is affecting how you design clinical trials, what clinical trial programs you can conduct, and so on, and so on and so forth. So that has slowed down our clinical trial progress in designing the protocols and things like that. That is natural with the situation right now. People are not necessarily available. People have to go into their self-isolations, and all those things are going on. They are affecting everybody. So in that sense, we are not losing time either, because the shingles program could not have started clinical trials today, even if we had been able to file an IND. So we are taking advantage of the opportunity that, or what you say, the obstacles placed in our path, into turning them into opportunities.
Absolutely.
So that's what we're trying to do.
So in terms of getting and really focusing on these indications that you are working on, COVID-19, shingles, of course, once that picks up back up again when you're designing the clinical trial, are you guys specifically really to the COVID-19 indication, are you guys looking for government support to once there's more data to read out to help push that along and fast track it in some sense? Or are you relying on all your own resources to push it forward?
So the way I think about it is that at some point we are going to need government support. Whether funding is needed or not, I do not know, because the way we are being a very novel technology, untested technology in terms of how they look at it. So because of that, we are going to have to do work on our own. Quite a bit of work. Antibodies are considered as a classical thing. Vaccines are considered classical thing. And here comes nanoviricide, they don't know anything about, right? So we have to develop much more greater data sets, I believe, maybe on our own before we can get to the funding thing. But at the point where clinical trials have to be organized and things like that, that is going to require government support. Without that, it is not going to happen. So yeah, but in terms of our own programs, we have the shingles program going on. It will continue, and we'll focus our programs into that as soon as this hopefully wanes down. Now, the other thing about the way we are designing our COVID-19 program is that we are not making a drug that is specific to SARS-CoV-2. We are making a broad spectrum drug against coronaviruses. And we have shown that at least we have seen that, I should say, and at least two very different coronaviruses. And so these are coronaviruses that don't go away. They are seasonal. They are coming back at us. And there are some patients that get serious disease. Most of us get common colds, but there are some serious disease cases always every year on these things. So that drug is also not going to be extinction type of a event for us. Right. It's going to be a drug that we can get and we can commercialize.
So this is, and so you're actually speaking to the release that you guys just put out. We're recording this on Tuesday, May 12th. So you're referring to the press release that you guys just put out today, correct?
That's correct. Yes.
All right. So, I think we've covered a lot today. My final question for you is, from what you can tell us, what would you say are the 1 to 2 things that investors should look for from the company now moving forward?
Looking from the company now, there are a few different things to look at. One is it's a platform technology. It's not a one-trick pony, and it is a completely novel technology. Like I said, we believe that we are making a nano machine to destroy the virus once and for all in the bloodstream. Sort of, I would like to introduce a joke here. Our president actually made a comment that if you could have a disinfectant that you could course through your bloodstream, you could really win against this battle. We believe that we probably have done that.
Hey, your lips to God's ears, okay? I mean, listen, I've done a few interviews now with a few companies that are working on treatments, vaccines, and all I can really say to each one is good luck, right? Yeah. I mean, because this is critical work right now, and we want you to succeed. Really, we're not just, this is, take away the capital markets and stock and all that. I think we can all agree. We just want to see success, but at the same time, we want to make sure things are being done correctly. And we're still so early. I mean, we're so early, So with that, Dr. Diwan, where can my audience go and find more information about NanoViricides and to continue to follow along the progress?
So our website, which is not great, but it doesn't get redone very often.
That hasn't changed in many years, actually.
But because that happens to be my lowest priority, I tend to spend more time in the lab. I need to get the company into the platform, into the regulatory affairs. And once that happens, then we can do other things.
But still, they can go to nanoviricides.com, right?
Yeah, yeah. And the press releases that we have are more talkative. Our case used to be very, they used to have a lot of information. Over the years now, we have started cutting down the information because we are focusing on herpes viruses alone. And so we are taking a lot of information. But there is that. And then basically they can send emails, info@nanoviricides.com. info@nanoviricides.com. Those emails come to me and a few other people, and most often they get responded to.
Well, Dr. Diwan, I really do appreciate you taking my questions. I know some of these probably weren't easy. It's been a long road. It's been many years of development, many different things going on there.
Much longer than what I originally expected.
I'm sure. So with that, I just want to say, stay safe. Thank you. Good luck. And I look forward to following along and for the next update. Okay.
You too. Thank you very much. I appreciate it.
Thank you.